Hello, and thank you for joining us the 6th Annual Global Summit Kidney Disease Innovation Vox Populi. The time is now to prevent the cure kidney disease. This summit is hosted by the American Association of Kidney Patients in partnership with our allies from the George Washington University School of Medicine and Health Science. My name is Orlando Torres, and I am an AAKP ambassador from Puerto Rico. As a kidney patient for many years, I have experience with in-center dialysis, home dialysis, and transplantation. My professional background includes international company in the pharmaceutical, banking, and health field. On my days in the dialysis center, I found AAKP as a great resource for educate myself as a renal patient, and eventually I started to work locally in benefit of my fellow patients as AAKP advocate. This year, the summit theme Vox Populi was purposely and strategically chosen by AAKP for historical meaning, voice of the people. I am one of the people who has chosen to raise my voice in support to the greater care, choice, and innovation in kidney care. And I am one of the voices demanding the change of status quo of kidney medicine. Over 550 million individuals across the world have kidney disease. This number is increasing at an estimated rate of 5 to 7% annually. Every year, we lose thousands of innocent lives to preventable kidney issues and the lack of timely access to treatment. AAKP believes kidney disease is both a workplace and healthcare issue. The disease takes the devastating toll of human life and livelihoods. Most people understand that kidney disease left untreated and untreated can lead to premature death. But many are not aware that the impacts a person's dignity, ability to provide themselves and their families. Kidney disease and outdated treatment can lead to unemployment, underemployment, disability, and dependency. In 2019, AAKP declared the next 10-year decade of kidney disease. The time is now for kidney patient, consumer, healthcare professional, researcher, industry, and all people of goodwill to unite our voice and demand changes. We are optimistic we can and will save more lives, but to do this, we must demand new screw guideline and diagnostic, new biology, and new devices. Together, we can manage and solve this growing global public health crisis. The time is now to elect and appointed leaders worldwide to listen patient voices. Governments must remove the regulatory and payment barriers they create that delay information and prevent timely care access. If government fails to act, we must raise our voice and demand an end negative government determinants of health. Make no mistake, every nation is impacted by kidney disease and millions of people are in risk. The optimistic news is that every person watching this program today can help those impacted by kidney disease and make an impact with their own nation. Through AAKP Global, AAKP is educating patients, care partners, healthcare professionals, and policy leaders across the world on unmet patient needs. Our annual Global Summit of Kidney Disease Innovation has helped put a human face on what is now the 10th leading cause of death based on data from the World Health Organization. We have connected people across the globe in connection purpose larger than every one person or nation. Together, we have increased hope and optimism. So thank you to join us. Your voice is a voice of people, and you can count on AAKP to keep applying throughout the world to create a change. With that, I am honored to serve as a moderator for our session entitled Alternative to Human Organ Xenotransplantation. I am thrilled to introduce our next speaker, Dr. Leonardo Riella. Dr. Riella holds the prestigious Harold and Eileen Danser Endowed Chair in Transplant Surgery at Harvard Medical School and serves as the Medical Director of Kidney Transplantation in Massachusetts General Hospital. He is also Associate Director of the Legorreta Center for Clinical Transplant Tolerance and a Senior Investigator at the Center for Transplantation Science. Dr. Riella research focuses on understanding immunoregulation mechanisms and developing new therapies to promote immunotolerance. His lab is at the forefront of addressing the organ transplant shortage by exploring innovative approaches such as bioengineering kidney from human stem cells and utilizing gene-modified pigs as a potential kidney donor. This groundbreaking method have the potential to revolutionize the transplant field and significantly increase the ability of organ. Dr. Riella, I turn over you.
Thank you so much for the invitation, AAE and George Washington University. It is my pleasure to be part of this Global Summit 2024. My name is Leonardo Riella. I am the Director of Kidney Transplant Program at Mass General, Harvard Medical School, and I'll be talking to you the next 20 minutes about innovations in kidney transplantation, current perspectives, challenges, and pioneering innovations. This year, we celebrate 70 years since the first kidney transplant that happened here in Boston at the Brigham and Women's Hospital. Since then, we have seen a major success, and transplantation has become the treatment of choice for patients with end-stage kidney disease. However, there are still challenges. One of the major challenges that we have in kidney transplantation is the rejection of the organ. In order to reject an organ, the immune system unfortunately recognizes in the transplanted organ as a foreign body. So each organ comes with a unique fingerprint that causes the immune system to then be activated and attack the transplanted organ. So in order to avoid a patient to get a rejection after kidney transplantation, one must take immune suppressive drugs, anti-rejection medications that weaken the immune system. These medications act in the machinery that's required for the immune system to get activated. And we use different combination of medications together in order to fully shut down or inhibit the immune system in order to prevent rejection. The results are, if you look in the short term, excellent. So the 3-year outcomes after kidney transplantation at MGH are 96.7%. Nationwide, around 89.9%. So both excellent results of kidney transplantation up to 3 years. But there are problems in the long term. Most of the problems are related to side effects associated with the anti-rejection medications. The immune suppressive drugs, by weakening the immune system, can cause your immune system, your body, to be more prone for infections and cancer. But also, these same immune suppressive drugs have associated side effects such as high blood pressure, osteoporosis, diabetes, and even kidney toxicity. The immune system is in particular a very important piece that is significantly affected by the anti-rejection medications. And the way to think about it is that our immune system is our army. It's our army that defends us against any potential threat, being that an infection or being that even a cancer. When we weaken the immune system, we make our body more prone to have some of these threats taking over our own body. So what I do in the lab as a physician scientist is to study novel ways to control the immune system, to minimize some of these side effects. And we do this both in preclinical models, in animal models, as well as in humans, and we get human samples in order for us to better understand. And this is the cycle of science that hopefully will bring innovation to our patients who need the most. I'm going to have to focus, since I only have about 20 minutes, on two major challenges that I see in transplantation currently. The number one is overcoming this immunological barrier of transplantation, the rejection process. And two is dealing with the organ shortage that exists currently in transplantation. Let's start with the immunological barrier. There are multiple strategies for us to do a better way in suppressing the immune system by still allowing the immune system to fight infections and cancer. These are some strategies that have been used to promote immune regulation, teach the immune system to control itself and avoid the rejection process, including drugs that may block co-stimulation or enhance co-inhibitory pathways, the cellular therapy with regulatory cells that may help control the immune system, the administration of drugs that enhance that immune regulatory arm of the immune system, and tolerance through the combined bone marrow kidney transplant process, which is also called mixed chimerism. I won't have time to go through all this, but I'm going to focus on one, which is the tolerance. So why do I think tolerance is important? And maybe even before that, what is tolerance? So tolerance is the process in which there's an absence of destructive immune response against the transplanted organ without the use of anti-rejection medications, while at the same time maintaining an intact immune response against other threats, such as infection and cancer. There's only one way that we've been able to develop tolerance in transplantation so far, and that's through a process called mixed chimerism. This is when we do a bone marrow and a kidney transplant at the same time. So you're going to have the donor immune cells interacting with the recipient immune cells on a number of different secondary lymphoid organs, such as the thymus, which is the school of the T cells. This allows this interaction of the donor and recipient immune cells to re-educate the immune system and tell them not to reject the transplanted organ by deleting and destroying cells that may recognize the transplanted organ as a threat and enhancing cells that may regulate that immune response. The process of tolerance through mixed chimerism involves basically a preparation of the body to receive the bone marrow, one irradiation to the thymus, which is this school of the T cells, and then on day 0, instead of just getting a kidney transplant, you also get a mini bone marrow transplant. So you get some cells from the donor bone marrow that are going to be crucial on that initial phase to re-educate the immune system. Within 1 month, patients are only on one anti-rejection medication, and after the first year, if everything goes well, they can stop completely all immune suppressive drugs. There has been a number of patients that have gone through this protocol. This is one of the first patients that got a first transplant back when she was 12 years old, had a number of complications, including viral infections involving her skin. And after her second transplant, she decided to go through the tolerance protocol. She got both a bone marrow and a kidney transplant from the same donor, and she has been now immune suppressant free for more than 17 years. I have an example here of a second patient, similarly, that got a bone marrow and a kidney transplant at age 36, and now she's 50, immune suppressive free, without any of the side effects of anti-rejection medications. So this was published back in 2008, which was the first kind of case series coming from Massachusetts General Hospital. But since then, we have been able to optimize even further the protocol, making much less tolerable for patients. And this is the revised protocol that was started back in 2022 that also involves a very similar pattern, but much less drugs given early before transplantation in order to prepare the body to receive the combined kidney bone marrow kidney transplant. And then following transplantation, patients are maintained solely on tacrolimus and then withdraw after 6 to 12 months if everything goes well. This led us to launch the first clinical transplant tolerance center in the world. So this was the opening ceremony in 2023, last year, that allow us now to create a team dedicated to this protocol. It's not easy to do the mini bone marrow and the kidney transplant, but if you are in a center that has the expertise, our results have been extraordinary. And these are some of the inclusion criteria of patients that may qualify for this. Ages between 18 and 65 at this point. They are either planning to have a kidney transplant and have a living donor, or had a prior kidney transplant with a living donor in the past. So there's possibility of doing the combined bone marrow kidney at the same time, but also doing the kidney transplant as a first step, and then following a year or so, doing the bone marrow transplant in order to induce tolerance to come off all anti-rejection medications. I have some barcodes here with some of our websites with additional information if any of you are interested in looking at it. So on this first phase, what I wanted to describe to you is that clinical transplant tolerance is the ultimate goal because it minimizes and eliminates the requirement of immune suppression in transplantation. Second, that MGH transplant tolerance work is based now over 40 years of research and really changes the paradigm. Instead of suppressing the entire immune system, let's re-educate the immune system in order to prevent rejection. And the current protocol that we have since 2022 really minimizes any toxicity and improves the opportunity for patients to go through a tolerance protocol in kidney transplantation in general. So we discuss overcoming the immunological barrier of transplantation. Then the second topic that I'm going to briefly coverage is the major organ shortage that we have in kidney transplantation and options for us to overcome this. So there are more than 100,000 patients on the waiting list currently. However, there's only about 12,000 kidneys a year, just not enough kidney to give to these patients that are in need. Not only that, if you look at just at the regional level, you can look here in the middle where MGH and look at over the years, how our waiting list grew to more than 1,373 patients in 2024 waiting for a kidney transplant. What is unfortunate is that many of these patients are going to have to wait on dialysis. While dialysis is an amazing treatment that can keep patients alive, the longer you stay in dialysis, the more damage you may have to your cardiovascular system. So just to have an idea, the risk of dying on dialysis is 6 times higher than if you get a kidney transplant. Not only that, dialysis also significantly impacts the quality of life of patients. So we see dialysis for patients that are candidates for transplantation only as a bridge to get them to go through a kidney transplant. So there are a few strategies that can be that have been worked on and researched in order to minimize and overcome this organ shortage. One of them is bioengineering of kidneys. So this is the creation of kidneys in the lab. The second one is xenotransplantation, which is creating compatible kidneys from other animals in order to reduce this organ shortage. I'll start with the bioengineering of organs. So what we've learned over the past 20 years is that we can get stem cells, and with a combination of nutrients and specific factors, we can create all mature kidney structures that are required for a kidney to work. And what you can see here with all these different colors is all the different parts and cells of a mature kidney that can be produced from only a single stem cell. Not only that, you can also produce the draining system, the pipes that form the kidney, and this is the ureteric bud, and you can clearly see the potential of creating these mature cells, putting them together, and creating a fully functioning kidney. And this is one of the projects that we've been working now for the past 5 years in collaboration with the Lewis and the Morizani lab with support from the Humans and Organs and Physiology Engineering program. And what we do is thinking about growing these stem cells into mature kidney organoids, and then using a bioprinting technique in order to create vasculature and optimize the survival of these organs and ultimately perform the filtration required for a kidney to work. Let me just show you the two major challenges that we have been able to overcome. Number one is to grow enough of these kidney organoids, and we developed a technique that involves a bioreactor that allows the growth of these kidney organoids in large volumes. The second piece is the bioprinting, so the bioengineering of channels that allow nutrients actually to go to the cells. And what you can see here is how this bioprinting kind of work. It's called the SWIFT technology. It's almost like a needle that creates this simple channel here in this case, but you can create all sort of complex channels inside that kidney organoid in order to reproduce the vascularization of the tissue and prevent any problems with nutrient delivery. And then you can see here on the right side how this channel kind of looks, and you can re-endothelialize the internal part of it to prevent any risk of clotting. So after that, and just moving forward, I won't show you all the details. We put this together in a structure that looks exactly similar as a kidney with a vein or an artery and a drainage system. So this is being implanted into mice. And what you can see here is the technique that we use. Mice have very small blood vessels. We use a cuff technique. This is the inflow of the artery. This is the outflow of the vein. And what you can see here on the right side is a mice after the anastomosis and after you've seen the blood flow going through this mini kidneys that had been bioengineering from stem cells. So there has been a lot of advancements, but there's still challenges. I think the number one challenge that also exists when you use stem cells is the rejection process, in particular if you're not using stem cells from the own transplant candidate. And here we show that the immune system response is very similar to what we see in a solid organ transplant in humans. And you can clearly see how all these genes get upregulated and cause the rejection process. And if you use anti-rejection medications, you are able to inhibit that. The second challenge that's still ongoing is how to optimize the filtration and the drainage system in order to fully recapitulate what happens in humans, the efficiency of the kidney and the filtration efficiency that allows to remove all the toxins and so forth. So this is an evolving field, I think has a lot of potential, but there's a few more challenges that we have to overcome in order to bring this to our patients. On the other side of the coin, we do have a specific innovation that has already approaching patients, and that's xenotransplantation. So xenotransplantation is the transplant of an organ from an animal into humans. And this had a number of different challenges over the past 40 or 50 years in which xenotransplantation has been considered. The number one challenge is that these humans have autoantibodies, have natural antibodies that reject the transplanted organ from an animal because of the presence of these pig sugars, in particular in the case of porcine donors. We also have some differences in our complement and coagulation system, the potential risk of infections, as well as a very strong immune response that the human immune system creates against transplanted organ from an animal. But fortunately, over the past like 15 years, there has been a huge advancements in creating kidneys that are more compatible to humans. And this was possible with the use of the CRISPR gene editing technology. The CRISPR gene editing technology, it is a specific tool that allows us to manipulate the genes in a very, very precise manner. So we're able to remove things on the pig kidney that make less compatible to humans, such as the pig carbohydrates. We can also inactivate viruses that we don't want to carry any risk in humans. And we also can add certain genes and proteins that may allow us to be more compatible in terms of the coagulation cascade. And this was a partnership that we did with eGenesis by creating this gene-edited pigs to make them more compatible to humans. And with those pigs, we have shown the best results possible in non-human primates. The research has evolved. So the first xenotransplants that have been done more than 20 years ago only lasted a few days. And now we have kidneys lasting more than 2 years in non-human primates. And this was just published last year. With that data at hand, and knowing that these transplant xenokidneys can perform all the essential functions of a normal kidney, including the water and mineral metabolism. And knowing that, these kidneys can last more than 2 years in animals, and that short experiments in humans, in particular human brain dead donors, have shown that there's kidney function and no immediate rejection, allow us to feel confident and justifiable to move xenotransplantation to the clinic. And this is also important in terms of thinking about safety. So these animals are grown in a very isolated fashion. So there's they're not close to other animals. They're monitored very closely, testing for a number of infection pathogens that the FDA had determined. So overall, the risk of transmission of an infection from a pig to humans, in particular those pigs that are grown in a very isolated manner, are extremely low. This led us to approach the FDA to submit what we call a compassionate use application. So this is a regulatory pathway in order for us to get a treatment that's not approved to be used in humans, but to be used in a patient with a life-threatening condition, so that we can better understand and potentially advance the field going forward in the design of clinical trials. And the way we selected the first patient, we were looking for transplant candidates at our MGH site that were ongoing dialysis, that had failed a prior transplant, that had no living donors, and they had a waiting time of more than 4 years. And also they were having a lot of challenges with dialysis, such as vascular access. And so their prognosis were very poor, meaning the chances of them waiting those, say, 5 to 6 years to get a human kidney transplant was close to 0. And in March 16th, 2023, we performed the first xenotransplant into a living human being. This is the surgical team that was involved, led by Dr. Kawai, here shown on the left, who has one of the greatest experience in xenotransplantation in the world, in particular with the non-human primates. And the process of the xenotransplant, it's very similar to a human transplant. So there's first the step of removing the organ from the donor, in our case, our humanized pigs. Once you remove the organ, you prepare the organ to be transplanted. You transport it in ice to the hospital in order to get transplanted and operated on the recipient. The kidneys are connected, our artery and a vein and a ureter, which is a drainage system. Once the perfusion of the kidney is done, you see how they change color. So on the prior one, without blood, the kidney looks pale. Once he perfuses blood, it becomes this dark red color. And within a few minutes, this is the ureter, so the pipe that drains urine from the kidney, we could see urine being produced. And within the first 24 hours, the patient made more than 3 liters of urine, showing that the kidney worked right away. This is an imaging of the kidney showing the blood flow throughout the kidney, that this is how it looks normally. And this is what happened with the kidney function. The kidney function progressively got better from a creatinine around 8 to 9. It went all the way down to below 2. At the end of the first week, we did have a rise in creatinine, and we identified a rejection that was completely treated. And at the end of 36 days, right here, we did a 24-hour urine collection, and the creatinine clearance was 60 mLs per minute. And on day 44, the creatinine was 1.5 milligrams per deciliter. Very normal expected creatinine for any kidney donor recipient. What about the minerals, which is one of the most important pieces of physiology that the kidney is responsible to? We can clearly see how the potassium has remained within normal limits across those 42 days here. What about blood pressure? We know the kidney is also important in regulating the blood pressure, and we can also show here that the blood pressure was stable throughout the course of the post-transplant period. Of course, the major hero in this whole story was our patient. So he was discharged home 20 days after the xenotransplant. His hemodialysis line was removed, and it was a hemodialysis line that he was having a tremendous amount of challenge, and all his electrolytes were stable, and he had good urine output, and his latest creatinine was 1.7 milligrams per deciliter. Unfortunately, about on day 52, after being seen in the clinic, he went home and had a cardiac event, ended up passing. So despite the success of the kidney, we were able to analyze and prove that the kidney was functioning normally. There was no evidence of rejection, but unfortunately, his heart was not in such a good health based on his long years of dialysis prior to getting a kidney transplant. So xenotransplantation has really the potential to address the organ shortage. I'll show you that a xenotransplant can preserve a good kidney function, maintain electrolytes with no evidence of infection transmission, and really moving to a small clinical trial is the best next step. There has been a huge team that was responsible for being part of this. It really takes a village in order to do something like this, and my greatest gratitude goes to our MG8 team, as well as our collaborators, in particular eGenesis, and our entire research team that allow us to really try to bring these innovations to patient, and when we have challenges, to go back to the lab and work on potential solutions going forward. Thank you so much, AAKP and GW, for the invitation to participate in this global summit.
Thank you, Dr. Leonardo Riella, for your informative presentation. Before we conclude, I would like to pose the same question to you as I did to Dr. Riesemer to get your perspective. First, there are any concern about future xenotransplantation kidney recipients being more susceptible to viruses like COVID-19 and bird flu versus the regular population of those with human kidney transplant?
Infection is a major concern in transplantation since we're using anti-rejection medications that weaken the entire immune system. So in order to compare human and xenotransplantation, what it is important to know is that animals are grown in a very strict environment, controlled and checked for any potential infections after transplantation. So we feel that the risk for transmission of infections from an animal to a human in the case of xenotransplantation are very low.
Also, will xenotransplant recipients be more likely to carry virus or be host to new COVID or other virus variants because they are immunosuppressed and have a weak kidney?
So transplanted patients do have a weakened immune system, making them more susceptible to any type of infection, being that bacteria or viruses. This infection can come from the outside or sometimes can come from a potential donor. Both in case of human and animals, we do an extensive testing of the donor prior to transplantation to minimize any risks. Therefore, the risk remains extremely low for any transmission of viral infection from a xenotransplant donor into a human recipient.
Can you talk about how different countries plan to regulate xenotransplantation research and clinical trials?
I can talk a little bit about how the US is regulating xenotransplantation. The FDA is one of the major regulatory agencies that's overseeing the potential use of xenotransplantation in clinical trials in human. And this is being very rigorous study for a number of years, and most likely will be a very stepwise approach in order for us to get into clinical trials in which humans could be enrolled. Other countries may have other types of agencies that are overseeing it, so it's very hard to speak about those. But in general, we in the medical community feel strongly that it is very important for us to do this in a very regulated fashion in order to minimize any risk for our patients. So safety should be the number one rule for us bringing xenotransplantation to the clinic.
Our next question, can you discuss the potential for xenotransplantation to be integrated into the standard medical practice in next decade? What new system will be needed in terms of A, the hospital system, B, transplant center, C, post-transplant follow-up care?
Transplantation is a very specialized field. And when we talk about xenotransplantation, the degree of expertise is even higher. So I foresee that xenotransplantation will be initially concentrated in a few transplant centers in the country. This is because we need to learn the most and have all the expertise possible in order for this to make it successful. Once the process is optimized, we know exactly potential complications and concerns or problems that may arise. It will be easier to then dissipate this knowledge to other transplant centers to make it a much more diffusely available process for our transplanted patients.
And finally, how might xenotransplantation impact global health disparity and access to transplant services?
Transplantation, similar to other parts of healthcare, has an enormous disparity. We do know that patients coming from underserved areas are less likely to get access to transplant and less likely to be transplanted ultimately. Potentially, xenotransplantation has the option of serve as an alternative source of organs for some of these patients that may not have a living donor in order to get transplanted right away. So it's very important as the field of xenotransplantation evolves that we think about how can we use this innovative field to minimize disparity across our transplanted patients.
Dr. Reynaldo Riella, thank you for sharing your time with us and the thoughtful presentation on the future of transplantation.